supérieur d'agronomie, Montpellier. And with this stage, he graduated with a PhD student in systems dynamics. Then I was really curious to know that he was one of the first employees of the Max Planck Institute of newly founded Max Planck Institute of Dynamics and Complex Systems in Magdeburg, where he established the Department of Systems Biology. He graduated in 2004 and in 2005 became an assistant professor in bioinformatics at the computer science department at ETH, where he's now leading the group of and the department DBSC here in Basel. And what was really interesting is that was when I was looking at your research activities, you have an extremely heterogeneous interest. So basically, your group is covering pretty much everything from synthetic biology, theoretical models, mathematical, computational, dynamical systems. So I'm really looking forward to the talk today, and thank you very much for giving a talk.
Thanks a lot for this very kind introduction and also for the invitation here. Today I decided to focus on just one aspect of what we are looking at in our heterogeneous environment, and that's on cell-to-cell variability, but in a slightly different sense than it was presented today. The examples will be drawn from yeast biology. And I already did a great job in introducing yeast and why it's important, so I'm not going to talk about this. But more specifically about the topic. So what we're talking about is phenotypic heterogeneity. So the assumption is you have a population of cells, they are genetically identical, and they also live in the same environment. But still you can have different behaviors that are typical for each individual cell. Yeah, so there are many possible causes for this phenomenon. So it could be stochastic noise, it could be some other sources, it could be cell cycle stages, it could be different cell ages, etc. And the aim, what we want to understand is using mathematical modeling, what the causes of this phenotypic heterogeneity are. And the reason why we want to do this is, on the one hand, to understand fundamental biology, like, for instance, how does population dynamics, bed hatching, and other phenomena, how do they actually work in real systems, but also in order to try to use or exploit this type of variability in order to assign new functions in biology, so synthetic biology. So that's what connects these two topics. So to go a little bit more into detail, so we start from the experimental data. We look at a particular type of single cell analysis. So this is all live cell imaging. So here we focus on this green yeast cell. They can be grown over essentially several weeks now in microfluidic chips. And what you get at the end is both for this particular cell here, fluorescence time course information in real time here, as well as for the entire cell population, you get lineage information. So you start with this one cell here. This is the green one that is highlighted. And then they see the lineage tree. So this is the type of quantitative single cell data we start from. Just as a little bit of a side, so this movie looks nice, I hope. There's a lot of infrastructure behind this and generating this here, and this is mostly on the side of software. Yeah, so what we try to do, and this is collaboration with many different groups within ETH and outside, for instance, having all this data generation, acquisition, metadata information into one laboratory information system integrated. In order to do more advanced experiments on the microscope in real time, we developed a software, microscope control software that's called UScope, that allows you to actually do experiments such as doing very dynamic time courses, including, for instance, changing media in real time, etc. And then the other aspect that was important was to get highly accurate image analysis. So we start from these images here for the population of cells. In the end, we want to have each individual cell segmented and doing this in a very precise way. So if you're interested, all this software is available in this open source. So once we have this data acquired, the question we're going to ask is the following. We want to know what the reason for this heterogeneity is, and I'll just give you two possible explanations here on the left. So one is simply that you have stochastic effects, so low molecule copy numbers and the probabilistic nature of biochemical reactions may be the cause for these individual cells here, so these individual trajectories here behaving differently. The other possible explanation is that the cells per se are in a different state, so this is usually subsumed under extrinsic variability. Yeah, so the processes are external, like cell cycle regulation, could be cellular resources, could be cell age. So that's the experimental data. What we want to understand is now for each individual cell here, which of the two processes describes best, or these two hypotheses describes best the real observed behavior. So this is quite a challenge, and I want to illustrate this with a few examples. And so look at this simple pathway here. So we have protein production and degradation, just two parameters. K here is the production constant, G is the degradation constant. And these are just simulation results. So here in this scenario, we assume that each cell has an individual set of parameters. So there's values for Ky and Gy. And you get population heterogeneity because they behave differently. And you see the distribution of these two parameters here in this plot. Okay, but otherwise the dynamics of each individual cell is assumed to be deterministic. And you will find the deterministic mean and essentially these trajectories here, one is highlighted here. This is just a result of observation noise, measurement noise. On the other hand, you can do a simulation like this, stochastic simulation here. Now the parameters are fixed. Every cell is assumed to have exactly the same parameter values. And you get variability within the population just due to stochastic noise. Okay, now if you adjust the parameters and the variability here in a correct way and look at one time point, take a snapshot, look at the distribution over the population. So this would be the density of the protein molecule numbers here. They are indistinguishable. Yeah? So you can explain, at least with this data here, with the snapshot data, you can explain this data either way. You cannot decide. It gets a little bit more interesting when you take a slightly more complex model. So this is now an extension. So we have, again, this protein Y here that gets produced and degraded. But now essentially you have a positive feedback by this component X, which effectively makes the system a bistable switch. Okay, so same idea. We look at varying parameters. We do deterministic simulations, ODE systems here. And you get several trajectories. You see that they are still heterogeneous. But now the interesting aspect is if you look at the average of the population, that's the solid black line. Just take at each time point the average of the data. This is different from what an average cell would do. So an average cell is the cell that has the average parameterization here. Okay? So this is due to the nonlinearity of the system. You see the same phenomenon when you do this for a stochastic case. Okay, and the final complication is the following. So far I showed you one distribution, so this one here on the left, for how these two parameters are correlated. Essentially, they just vary in one direction. So this simulation here has exactly the same average. It has exactly the same variance in the parameterizations, but they covary differently. So here they're uncorrelated, and here they're highly correlated, and you see this has huge effects on the dynamics that we predict. So even if the average cells are identical, even if the total variance is identical, you can get very different behaviors. So this is now what essentially the problem statement is, what we're trying to do. And this is all summarized in this recent paper here. First of all, we want to try to infer a mechanistic process model from these dynamics of single cell data. So eventually trying to figure out which causes there are for this cell-to-cell variability. And this is done via nonlinear mixed effects models that I'll introduce in a minute. And the second task is then to try to figure out which of the processes, what in the biology actually contributes to the variability that we observe. And there I'll give you a proposal that involves global sensitivity analysis. So for the first task, The concept for these nonlinear mixed effects models is the following. So we formulate a dynamic model for each individual cell I. The hypothesis there is that each cell behaves completely deterministically, but each cell has a different set of parameter values and initial conditions. So that's what is heterogeneous in this population of cells. We have another element of uncertainty. That's the measurement noise. We assume we don't know what the noise is, so that's something that we have to estimate as well. And then we have to relate the individual cells to the population itself. And the hypothesis is that we can infer distributions that give us the relations between these individuals and the population. So that's the basic idea of NLMEs. A little bit more formally, it looks like this. So here we have an ODE model. Xi are the states for cell I. That depends on parameters alpha, depend on initial conditions. We lump all of them together for each cell. So beta I is the parameterization in terms of initial conditions and parameters for cell I. So that's the first level, so the level of the individual cell. We also have the measurement model here. So that tells us what yi are things we can observe. Xi are in the internal states, they're related. And there's measurement noise here. And then the second level is the level of the population. And here we assume that essentially that these beta I, the individual cell parameters, are generated from a distribution that has an average. So this is called the fixed effect or the population average, and some distribution around this average. And a bit for the notation, so this is this population average parameter here beta. This is the covariance of the individual cell parameters characterizing the distribution. So the reason why this is quite challenging is the following. So first of all, this problem scales quadratically with the number of unknowns, essentially with the beta C we're looking for. Because we have to estimate these covariances, so that's a square, it's a quadratic matrix. So most of the methods, and they come from pharmacokinetics, pharmacodynamics primarily, they can do this up to 40 effective parameters, roughly. But this entire approach is not only used in pharmacokinetics, but it's also extremely important there, and we're trying now to do this for single cell analysis. So just to give an idea how this works for a practical example, that's our benchmark example. So that's from work from a group in Paris. What they did was stimulate yeast cells with osmotic stress. That I already included this in our presentation, so that's the input, and then they just have a reporter gene that generates fluorescent protein here. So that's we apply this externally, or they applied it externally, and then just recording the response here. So the model here on the right-hand side is very simple. It just has 4 parameters, like here, the input is being sensed, then we have a parameter for transcription, the mRNA degradation, translation, and protein degradation. Okay, so in this NLME frameworks, this is roughly 20 parameters that need to be estimated, so 4 squared plus the 4 original ones plus a few noise parameters. So existing methods trying to tackle this problem for this particular example, there are 2 main ones. One is called the naive approach. The idea is you take each individual cell, you have data for each individual cell, estimate parameters for each cell, and then take the averages and variances directly from these estimates. The problem with this here, and this is illustrated in this slide here, so that's this NTS approach. Here's the real data in black, and the variance around the new real data. You see that this estimate is very bad. So it doesn't reflect the data at all. And if we just look at one parameter, here this is the distribution, the estimated distribution with this approach. This is bimodal, and it's far from the true one. So what is the state of the art? It's maximum likelihood estimation. So many pharma companies also use a particular commercial implementation. There's a type what's called monolix. What it does, it estimates all these parameters jointly. So that's the blue curve. And you see that the estimates here for the mean are really nice, for the variance or the confidence interval are very nice, and you get one parameter distribution here. Okay, so the problem here is this scales pretty badly and is a very costly computation. So what we proposed in this work is now the following idea. We propose a two-stage approach, but it's called global two-stage approach. So we have data for each individual cell, we fit each individual cell, and then we pool all these estimates. This is the first stage. What we then do is try to estimate the probability distribution that we need in the end, so the population level parameters, the estimates here, by essentially shrinking these individual samples here. So the intuition behind this is that here we get estimates for the parameters that are uncertain, and we take this uncertainty into account in order to come up with a formulation of the distribution that we finally use. So just to give you an idea of how this works algorithmically a little bit in more detail, so we formulate log likelihood that we want to maximize. So what we're looking for are these parameters, so the average average population parameters, the covariance. We have the individual covariances, that's what we get from each cell estimate. And then we essentially do an EM algorithm. So we first refine the cell-specific parameters, and then do the maximization step in two steps, essentially getting the population average for these parameters here, and then for the covariance matrix here. Now it stopped. Okay, if you do this, just to show you what the outcome here is, this is now the red line. You see the estimates here for the mean are close to the experimental data. For the confidence intervals are close to the experimental data, and also they overlap really nicely with the state-of-the-art algorithm. Now, when we just do in silico studies with this particular model and data set, we first asked, okay, what's the computational time we need in order to do this estimation as a function of the number of cells we include? For SAEM, this goes up extremely quickly. So this is all in parallel computing environment, whereas for GTS or approach, the slope is much slower. What was interesting in this analysis as well, this is a measure of the accuracy of the estimation. So 1 means perfect estimation. You need very few cells in order to do this estimation for this particular example. So in this case, around 100 are sufficient. And also the methods can tolerate a lot on whether the cells are mistracked in the image analysis or whether the tracks are not complete. So that was surprising. Okay, to go to a larger application example, that's a part of the analysis we did in our own lab. So the biology is that we have an amino acid transporter in budding yeast. So this is called MOP1. It's just labeled with a fluorescent molecule, so GFP. And what happens when you cultivate yeast cells in normal media without methionine, so MOP1 is the transporter for methionine, up to this time point 9 minutes. Then they express this transporter because they use it as a high affinity importer of the amino acid. But when you add methionine at this time point 9, you see that the transporter is being internalized. It is endocytosed. You see the spots here, and overall, the protein is being degraded. So the type of data we get from this image series are essentially the intensities of fluorescences in different compartments, like in these endocytic compartments or on the cell membrane where it's usually being expressed, or in the cytoplasm. So here's the real data. So this is the mean. These are individual cell trajectories, and that's the 90% confidence interval. And what we then asked for this particular example is, we see cell-to-cell variability, and what are the causes for this observed phenotypic variability? So the model we developed for this particular network is relatively simple. It's illustrated here. So essentially we have this protein MOB1 produced here by transcription translation. Then it's being transported to the membrane, where it usually acts as an amino acid transporter. But then it can be undergo endocytosis, it can be recycled, and it can also be degraded. And the input, namely when you give external methionine, what it does, it inhibits the production and also mediates rapid endocytosis of the transporter. So this looks like a small model, but it in fact has 14 kinetic parameters, 4 initial conditions, around 200 parameters that need to be estimated effectively. And with this GTS method that we proposed, this actually seems to work pretty nicely. So again, here these are the different observables, fluorescence over time in different compartments for single cells. Black is the data, gray are the sample trajectories. And these are now predictions. This is not the estimation, these are predictions of the behavior. And if you look at GTS in red, then this behaves nicely, also in terms of the variances. The SAM, the more complex algorithm, actually has difficulties, so here it diverges. Yeah, so we can consistently predict these single cell behaviors. So the result of this estimation are parameter distributions, like these ones here. So these are the 14 parameters. You see that the shapes are not necessarily normal, so we don't make any assumption on normality of these distributions. But these parameters cannot be directly interpreted. So these distributions here. And the reason is that they are correlated with each other. So this is shown here. So this is derived from this covariance matrix and shows you the correlation between the parameters. And you see here, of course, on the diagonal, they're correlated, but you also see that some of these parameters are extremely negatively correlated. Some are positively correlated to a significant extent. So the next question that we asked is now, how can we try to map these estimates on the parameters back to the biological processes and then figure out which processes are responsible for the variability. So the idea or the proposal for how to do this is the following. So this is something that is called first order Sobol indices for parameters. So it's a type of global sensitivity analysis tool. Just to give you a little bit the idea. So what we're looking at is the variance here for one parameter. So we look at the variance with respect to parameter j and the expectation with respect to all parameters but j. So what we're asking is how much of the variance being explained by variance in parameter j. This is our observable or our data. This is our parameter. And we do this with this expectation with respect to all the other parameters. And this is what we get from our estimates here. Yeah, so this is just the fraction of variation explained by this variation in this one parameter. So to show this for an example, how this works. So this is again this switch-like small network. For this example I showed you before, so this is the behavior of the OD system. Here there's just the variance in one direction, one parameter dimension. And the output is shown here of this analysis with the Sobol indices as j. And this means the green dots are at 1, means this parameter, in this case, this interaction here fully explains the variation in the data. Yeah, and we only have variation in this direction in here. When you have something like this here, so variation in more than one dimension, this tells you, the analysis tells you initially, it's this process that dictates the variation mostly, but then protein degradation starts to become important. And in the third example, when they're completely correlated, they can both equally well explain the variation. So essentially this means the parameters are not identifiable. Okay, now back to our endocytosis, what this analysis yields. It's a bit complicated figure here. So these are all this is the model again, the corresponding processes, and the parameters. They're colored by groups according to how they explain the variation. So this is essentially based on the correlation matrix. We build clusters of parameters that belong together, like these green ones here. So you can directly see they have to do with the production degradation of the protein in the cytoplasm. The numbers here and the size of the squares indicate how much variation there is in each of the parameters. For instance, this one is one that is extremely noisy. And these here are the Sobol indices. Okay. You see for certain things like cell volume, only these parameters here are important. So these are growth parameters. For other observables, for instance, what happens at the membrane, only these parameters here are important. And for some others, there are other groups of parameters that have importance. So that's the idea. We look at these sensitivities here, these first-order Sobol indices, which allow us to tell you which of the processes are important at which stage in the dynamic process. Okay, just to quickly summarize, I want to show you is that there are single-cell analysis. So when you have dynamic single-cell trajectories, they really pose new challenges both for modeling and analysis. The proposal here is to use these nonlinear mixed effects dynamic models, because we can include variation at different levels, at the individual cell, at the population level. And the real thing why this could be interesting is that we have the data that we need for this type of two-stage approaches readily available. So this is currently no problem to generate dynamic trajectories from a few hundred cells in a microfluidic device. What is really interesting, and also an additional difficulty, so these correlations, both for estimation and interpretation. And the overall idea is when we combine this NLME idea with uncertainty quantification, like with these Sobol indices, then we can try to go from individual parameters and how they vary within cells to really biological processes that are responsible for the variation. Okay, finally the acknowledgements. So this was work by mostly Lakshmi and Hans Michael and Fabian in my group, and funded by Systematics, among others. And with that, thank you for your attention.
So thank you very much for a very exciting talk. Do we have questions? I imagine yes. Maybe people are tired and need 1 minute to think about it. Actually, we have one, yes.
Thanks, Joerg. Very nice talk. So I was wondering how this could, how could you scale this up? So I was thinking if you were to have many readouts, but not necessarily in the same individual cells, how could the approach expand, either if you try to kind of map them together or maybe have some joined but some different readouts? Or
yeah, so what we're currently looking at is essentially combining this type of single cell trajectories with FACS data or something similar, where we know something about the distribution in the population but not the identity of each individual cell. And there are relatively straightforward ways for incorporating this, because essentially what we're predicting is the mean and the variance and essentially the complete distribution over time, and then we can ideally just formulate another distance to for say the FACS data and just try to minimize that. It becomes algorithmically a little bit more involved, but it's possible in principle.
But would it be like, but kind of unambiguous, or because you could think that? I mean, it
won't be unambiguous, because then also for this example I showed you with the endocytosis, there are clearly things that are not identifiable, where we cannot pinpoint this variation in one process and not just a group of processes. Okay. But we think that it will help in essentially identify more.
Okay, thank you.
Any other question? Actually, I was wondering, so it's a very interesting talk. It's the second time I see it, though. So I was wondering how computational intensive is to train this nonlinear mixed effects model. And actually, I was thinking about that. I was wondering when it is safe to disregard this complexity and to treat single cells as many people have been doing so far. That means like identical cells with same parameters and maybe some standard deviation for each parameter. So when do you think this is a safe approach?
So computational efficiency, so for the examples I showed you, the compute times are on the orders of less than a day, even for this endocytosis example. We didn't, so the student ran a larger EGF receptor model with 40, I think, parameters. It works, but it's computationally intensive. The other question is really an interesting one, because here we assume that everything is deterministic. Okay, so what we can say is we can explain a certain amount of the variation in the population without having to invoke stochastic noise. Okay, but it could as well be that you can generate the same behavior when you have a stochastic model and just one parameterization. Okay, so what we're currently working on is essentially optimal experimental design for this type of models, where you then would perturb one parameter or a distribution of parameters, and then in order to maximize the discrepancy between these two possibilities. But that's still in the works.
Okay, so actually that's an interesting comment. I was wondering now how we'll compare again if we do just stochastic simulation, as you're saying. You just disregard the deterministic approach and you treat each cell as like a fixed parameters. So first of all, well, the computational complexity, but also whether the results, which one is more accurate, which one do you think will recapitulate better the behavior of each single cell and the population behavior?
If you do a stochastic model with distributed parameters, but nobody can identify that. So, yeah, it's sort of pragmatic here. And also we looked only at systems where we have high copy numbers, essentially, of components.
Okay, any more question? Last question of the session and the day. Okay, so in that case, let's thank Joerg one more time. Thank you very much for coming.